It was really when I was thinking about doing a PhD actually. This was the time when I was just exploring different areas in the field, which was quite interesting at that time. And it was really cell death, so-called apoptosis, programmed cell death, which really I became very, very interested in. I was fascinated by the idea that when cells actually die, that this is highly regulated and really follows a program, yeah. Yeah, it's what I said already, it's really about DNA damage. If you think about DNA, which is basically the blueprint of life, every information in the DNA defines the whole organism and the health of the organism. So any sort of damage will affect basically the health of an organism or the viability of an organism. So, and cells have a lot of mechanism to maintain the DNA and keep it basically free of any sort of damage. And cells and whole organisms have a lot of mechanism to maintain this stability. And this is really the reason why we really try to understand this, how actually cell can maintain its genome and so the genome stability basically. So any defects in this of the genome stability will accumulate in mutations, and these mutations can then lead to cell transformation and then to the diseases. And most prominent is cancer. Cancer, the hallmark of cancer is actually it changes its genome basically in favoring of just growing wild basically. So this is really where the motivation is coming on, to study these sort of response pathways which are activated if there's any problem with DNA damage.
Okay.
And Fanconi anemia is one of these diseases. It is basically a genome instability syndrome. So these particular patients, they cannot detect a special lesion of DNA repair, and this is the so-called intercell crosslink. So this, they cannot basically deal with this sort of damage. They cannot repair it, so they accumulate this sort of damage, and this can result in cancer. This is exactly what these children born with this disease basically have very early in childhood. They develop leukemia, which cause these intestinal crosslinks. And here we found this gene RNF113, a uctin-E3 ligase. So if we knock this down, this ligase, we see increased sensitivity to intestinal cross-linking reagents. So this was from my side. Our collaborator in South Korea, Professor Ko, she identified it from she came from a different angle. She found that this ligase interacts with FANKD2. FANKD2 is a Fanconi anemia pathway protein, quite a critical one. It gets mononucleated and then somehow promotes the repair of DNA lesions. As I mentioned already, I think what we really wanted to get a complete picture how cells can recognize and repair lesions. And there's still a lot of genes and proteins out there in the genome, we have no idea what they're basically doing. That's why finding new players are quite important to get the complete picture of a response pathway. And this ligase RNF113 seem to be one of these candidates. And by studying this new protein in this pathway, we might even find a new player which is then maybe important in cancer biology as well, because you can think about cells which maybe don't have have a high levels of of DNA lesions, high rates of mutations, for example. So it would be maybe a new player also in cancer biology. I think the most significant finding is this very specific defect in homologous recombination. What I mentioned before, that this loading from this recombinase RAD51 onto sites of DNA damage, these seem to be very specifically for interstrand crosslinks, but not for other sorts of damage like ionizing induced double-strand break. I think it's a very early stage now. We just have some description where it might play and what kind of response pathways RNF103 is involved. But I think the most important thing is now finding substrates for this enzyme and really further understand what kind of pathway it's playing with. I think this is an important next step. But in terms of drug, if you think about a lot of cancer drugs are actually damaging DNA. One, a very prominent one, is called cisplatin. It's widely used in ovarian cancer treatment or testicular cancer treatments. And this drug also makes actually crosslinks. And what you can think about now is if you treat the cancer with this cisplatin, cells die. But if you make another deletion where you shut off a pathway which actually repairs this sort of lesion, you can sensitize these cells for the cancer drug cisplatin. So the idea is, for example, we can block now with a drug, for example, the catalytic activity of RNF113, which then should actually sensitize cells for treatment with interstrand crosslink inducing damaging agent like cisplatin. So you can think about combinatorial therapy, drug therapy, basically. And this is also what people investigate now these days a little bit to follow this up. So basically sensitize a cancer cell line, or some cancer cell lines might be intrinsically already more sensitive to crosslinks, and you can further sensitize them. Because for example, for cisplatin, there is a major problem of resistance. So these cancers respond very quickly to cisplatin. They die a lot, but after a while, the cancer is growing back because it's not sensitive anymore to cisplatin. But we can then maybe resensitize them by shutting off repair pathway, which are maybe upregulated in response to cisplatin. So there is definitely room there to explore, and there several bigger pharma industry are focusing on nicotine signaling and try to target these pathways with drugs. But so far there's not really a drug out there which actually targets